Hi, I'm Reshmi. I also work at UT Austin and Tom Yankelev's lab. And I'll be talking about our work to do some of the same type of predictive modeling, but instead in breast cancer patients using MRI data. So to start off, our general goal is to apply this mechanism-based model and use our quantitative MRI data, and we want to predict tumor response to treatment early in the course of treatment for these breast cancer patients. I'll walk you through the steps that we take to do this. So I'll start off with our image processing pipeline, and then we'll move into model calibration, and finally talk about response prediction. But to start off, I want to talk a little bit about the data set and the general framework. So some things to know about the I-SPY2 clinical trial is it's a trial of neoadjuvant therapy. So that's therapy before surgery for locally advanced breast cancer and includes multiple subtypes of breast cancer, which I'll get into later. And they use this adaptive randomization in order to get more experimental drugs through the trial in a way that optimizes the success of the drugs. So in general, there's a new patient enters a trial. There is some sort of randomization, so they either enter an experimental or a control arm, and that's just if it's standard of care therapy or an experimental drug. They measure the patient outcomes, so if they achieve pathological complete response or if they have some residual cancer burden. And then they update the predictive probabilities for the drug depending on the outcomes. At this point, a drug might be graduated from the study or it might be taken out of the study because it was unsuccessful. And then new drug combinations can be entered into the trial. And the parts of this that are important for our study here are that this is a highly heterogeneous data set. There are 10 different clinical trials for the patients or clinical sites for the patients we're working with, as well as those 4 different breast cancer subtypes, and the experimental and standard of care drugs that we're trying to model. So when a patient is initialized in the study, a tissue biopsy is taken and they measure the presence or absence of certain receptors, so estrogen or progesterone receptors, and then also HER2 receptor. And the estrogen and progesterone are grouped together into this hormone receptor, and then you have the HER2. So this is the general US population, and we have the highest percentage of HR positive, HER2 negative, and then triple negative is negative for all of these receptors. HR positive, HER2 positive is positive for one of the first group and also HER2 positive. And finally, HR negative, HER2 positive. So the patients that we're working with have good quality MRI data for 3 visits. But for this initial study, I'm working with a subset of patients that have either triple negative breast cancer or HER2 negative, HER2 positive breast cancer, or sorry, HER2 negative HR positive breast cancer. And these are all in the standard of care group because we're trying to model the control patients before we move into experimental therapies. As for the framework, so once a patient is initialized in the study, they receive a pre-therapy imaging scan, so that's V1 here. And then 3 weeks into therapy, and this is paclitaxel as standard of care drug, they receive a second imaging session. And then after the first cycle, so this is 12 weeks of paclitaxel therapy, they have visit 3 data. And then there is more, there's a second cycle of treatment following that, and additional, sometimes optional imaging scans and surgeries where they determined if pathological complete response was achieved or not. But for this framework, we're right now only interested in the first section of this. So what we're doing is we're going to use the visit 1 data to initialize. We want to calibrate to the visit 2 data. And then using our tumor forecasting pipeline, we want to make a prediction at visit 3 after treatment. All right, so moving into the image processing pipeline. First, we just have the raw data. So the 2 types of MRI we're looking at here are dynamic contrast MRI is the first one. So there is 1 pre-contrast scan, and then the contrast agent is administered intravenously. And they range from 5 to 8 post-contrast images, and these are spaced about 90 seconds apart. That's a fairly long time, and that's because these are all routine MRI images that are taken at clinical sites for these patients. So you can see the contrast entering and then washing out slightly. And then the second MRI modality you have is diffusion weighted or DW MRI data. And we have that for 4 different B values. B values reflect the strength and timing of the diffusion gradients. So at B equals 0, you can see are the lowest diffusion gradients, and then B equals 800 is the highest diffusion gradients, and also weights for diffusion more heavily, but the trade-off is that you're losing signal. The next step is for each individual visit, we need to register, we need to first align the DCE data with the DW data. So we first just do this field of view alignment, and this comes from the information in the DICOM header about where the patient is positioned in the scanner. For this particular patient, you can see that the field of view were pretty similar, but if you look at the before and after, you can see that the tumor is better aligned after with the DC in blue and DW in red. And then the next step is to do a rigid registration to correct any sort of movement or misalignment between these 2 modalities. Then we segment the tumor RI. So this is done independently at each of the 3 visits. And the algorithm we're using to do so is a fuzzy C-means clustering that iterates through and assigns certain probabilities for a certain voxel either being inside or outside the tumor, and that is applied to a manually drawn bounding box around the tumor. So you can see the outline on the left side for each visit, and then just filled in red so you can see it more clearly. So those are all the individual visit steps, but the image processing step after is to align between visits, because we need to be able to compare the breast geometry in order to use it in our model. So you see left to right visits 1, 2, and 3. At the top is before registration, but at the bottom is after registration, where we're registering visits 1 and 3 to visit 2. And we use a deformable B-spline registration, but here we're imposing a rigid penalty on the tumor RI. And that's because if we're allowing our tumor to change shape, then we can't have any confidence in making a prediction for the tumor volume or size. So you can see within those red borders, the grid is pretty even, but it deforms outside. And then this is just a 3D visualization of how the tumor is actually being aligned. So you see that it's aligned better after registration at the bottom than before. So the last part of our image processing pipeline is we have several post-processing steps. The apparent diffusion coefficient map comes from our diffusion weighted data. What you're doing is you're fitting the 4 different B values. So that's at 0, 600, 800, or 0, 100, 600, 800 to come up with a map for the ADC. And here below that, we have our different tissue segmentation. So we already have the tumor RI, but we also need to segment out the fibroglandular tissue in gray, and then the adipose tissue in that darker gray. And the last step is to get a map of the number of tumor cells. This is linearly proportional to some quantities based on the ADC map, but you can see that where you have higher ADC values, so that's more diffusion, you have a lower number of tumor cells, and where you have lower ADC values, that's where you have generally your tumor and it's more dense. All right, so moving on to the mathematical model. What we're looking at is a change in number of tumor cells, and we're doing this as a function of a diffusion term, a proliferation term, and a response to therapy term. We also do this mechanical coupling of diffusion so that we're incorporating the stress that the outside breast tissue exerts on the tumor. Our calibrated parameters are this diffusion coefficient. We have a local proliferation map, so that's at every point in space, and then a drug efficacy rate. In this case, we just have one drug, paclitaxel. We have certain assigned parameters that come from literature values, and then our variables. And the one I'll point out here is that the von Mises stress is assigned differently for the different tissues, adipose and fibroglandular, and then the drug concentration is estimated from the DCE data. Generally, our calibration framework, we have the drug distribution and tissue type that come from that DC data and our model calibration inputs. That's how we initialize our visit 1 time point. And then we have our number of tumor cells, which is our initial condition. And the visit 2 number of tumor cells is what we calibrate to using Leavenberg-Marquardt nonlinear least squares optimization. So here are inputs. And then these are some example outputs for one particular patient with our global parameters and a local parameter map. Finally, some preliminary results for the 18 initial patients we have. This is one example patient again. So at the top is the drug concentration. You can see it goes up with those 12 cycles of paclitaxel and then decreases. Below that is the total tumor cellularity over time, and then the tumor volume over time is at the bottom. And you can see we expect it to match for visit 1 and visit 2. That's our initialization, and then visit 2 if our calibration is good. And then at visit 3, this is giving a pretty good prediction where you see the measured in red matching up with the modeled in blue. I'll show 2 example patients. So for this particular patient, if you look at visit 3, even though it predicted the overall cellularity and volume quite well, we see that the total cellularity and volume are a little bit off. So that's something to look into in the future. Whereas for certain patients, we get a pretty good local estimate. And then just for an overview of the patients, on the left, you have the cellularity. On the right, it's the change in volume. And we are getting pretty good correlations between the measured and modeled values for the total values. Then just to finish up, here's a reminder of our initial goal. And the preliminary results do suggest that we can use this tumor forecasting pipeline to make pretty accurate predictions in this multi-site clinical trial setting. In the future, we are going to continue to model all 103 patients in this data set, and then we want to evaluate the prediction accuracy as a function of both breast cancer subtype and drug class. I'd like to thank my lab members and co-authors and our funding sources, and I can take any questions. Thank you.
One interesting feature I saw in the model was that in the kinetics of response to therapy in the patient where there was sort of a bit of a response to start with, what was happening in the model, and it seemed to be backed up by the next data point, was that if it starts to shrink initially, it's going to, it's looking on that tumor volume. Yes. Oh, right. Is that, I'm seeing the tumor volumes dropping even more as time goes by. Is that just something, the difference between like number and diameter, or is there some like, is something about this model saying that there is like an enhancement of sensitivity as time goes by, or like more drug is penetrating it when it's smaller or anything?
So
Just to look at, yeah, go ahead.
Sure. This isn't a trend that I see in all the patients. This is for this particular patient. And I think it has more to do with kind of what the distribution of cells is in this tumor volume, because what the model is doing is it's really only looking at the cellularity. And then we take that cellularity and apply thresholding to get the volume estimate. But that is something I'd be interested in kind of looking more into.
Yeah, I mean, it's like it's
Yeah.
you could have had no measurements in here, and the model is giving you stronger effects, you know, at like 70
Right.
to 90 days. So it's within your own model, it's doing that. So you have the framework to poke the interior workings of your own model and ask, why is it doing this?
Yes.
And it agrees with the data. So
Yes. Yeah, thank you.